All right. So welcome back to our, I believe, the final video of this unit anyways, which should hopefully be a pretty quick one here. We're just going to briefly touch on the topic of mutation. So all this time we've been talking about, okay, DNA is replicating itself. It's being transcribed, it's being translated, and we always have talked about this base pairing rule that these letters always match up with each other, and that this DNA code is extremely important. It's our instructions for making everything that we need. Sometimes things will happen, though, to mess up our code. We'll have some letters that are no longer what they're supposed to be. So that's why we're getting into mutations about what happens if we accidentally kind of mess up our DNA code and what those effects are.
Example,
mutation.
So
So mutations are, again, just changes in our genetic material. So when the order of our bases is somehow messed up, we call that a mutation. Luckily, this doesn't happen too frequently. If you think about how much we have to replicate our DNA over and over again, our body's actually pretty good at catching these mistakes when they happen. But they do occasionally kind of make it further along in the process, and that's when we start to see these effects. And so what can happen is we can have bases that are switched. So maybe it's supposed to be an A, but now it's a T, or maybe it's supposed to be a T and now it's a C. So if we switch the letters, we can insert or add new bases. So now all of a sudden you see in our example, we've slipped kind of an extra C into the middle of our code, or we can have the opposite where we remove some DNA. So in this one, we have a T now missing from our code. So all of these are going to have slightly kind of differing effects, but all of these we consider mutations because we've changed our original DNA to something new now.
And there's a video up here if you're curious on what causes mutations. There's a short video linked here if you want to watch that, you can.
And so what happens when we have these mutations? We have a couple of options depending on kind of what's happened. So again, our whole idea with this is that we're taking our DNA genetic code, we're making copies of it in transcription, we're taking that mRNA and we're using that in translation to make a protein, and we're using that protein somewhere in our body. So if we think about mutations, that small effect in our DNA kind of goes down the line until it's affecting our proteins. And so a mutation can have a couple of different effects. It could, one, have no effect. So even if the letters got maybe swapped or added in, maybe that's not going to affect things at all. If you remember our codon wheel right here, remember that many of our amino acids have several different codons that code for them. So maybe if we just mess up one letter, it might still end up with the same amino acid. So we could have mistakes that maybe don't actually do anything. We could also have mutations that actually enhance or change the function of our proteins. So occasionally, again, we have good mutations that happen that are actually going to make this protein function better, or maybe change its function. It's going to do something else. Or we can have the flip side where the function is going to be decreased, or it can potentially just altogether destroy the function, that this protein is now useless and it can't operate at all. So again, we have lots of different mutations that can happen and lots of different potential effects from these mutations.
All right, so we can use this in spelling, if you think about it. So we take an original sentence, time to dream, and we change one letter in the word time. We change the I to an A. So essentially we're substituting, and all of a sudden we have tame to dream. So not quite the same as time to dream. We could also switch things around and do things in reverse order. Emit to dream.
Inspirational.
Emit to dream. I just try so hard. All right, or we could just take letters out. You ever left vowels out of a word? Sometimes you can still get the word, sometimes you can't. So we take the I and the M out and we end up with Tito Dr. Eam.
That's my favorite one.
Another thing to point out is Miss Knowles still read it with the first four letters, then the next two, then what was ever left. Just like the mRNA and the ribosome are going to just read three letters, three letters, three letters. It's always going to go 3, 3, 3, 3, 3. It doesn't know where it originally ended or stopped. So
right. So it's just going to kind of back everything up to fill in the blanks where we took those other letters out. Or we can add letters. We can make it time at a dream.
I think that is an Irish name.
It's Tim.
I think it's time. No, because it's double I. So time at a dream.
Some call me Tim.
Some call me Team. We never know.
So that's kind of like a language analogy for this. But if we remind ourselves of transcription translation, what's actually happening? If we look at the top, this is our normal DNA sequence: AAA, ATG, CTT, CTC, and then we would transcribe that into mRNA. So we've got the base pair matches, and then it's translated into amino acids. So our order would be what do we got? Phenylalanine, tyrosine, glutamine, glutamine. And if we have a mutation, which we'll have to pinpoint down here, where is it? So that C is turned into a T. So now when we read the codons, the mRNA is going to be AAA instead of GAA, which is going to change that glutamine into a lysine. We could have an insertion, which is going to bump everything back, like Mr. Hamele was mentioning on the previous slide. Ms. Knoll was reading the words with the original spacing. With translation, it's always 3 letters, so that ribosome through the tRNA is always reading every 3 letters. So if we insert one, and now we have CCT, that's going to change the mRNA to GGA, which is now going to be glycine. And then the next 3 letters are going to be TCT, transcribed into AGA, translated into arginine. So with each of these, and we'll go through I guess the same thing happens at the bottom. I guess the point here that I want to make is if you change the sequence of A's, T's, G's, and C's in the DNA, that changes the sequence in the mRNA, which changes the way that the ribosome reads it and which amino acids are added. And changing the order of the amino acids changes the shape of the protein, which changes its job.
So let's take a look at this in a real life example in a real protein in humans. A mutation that, as Ms. Ripley said earlier, could be sometimes a mutation can enhance a function or change it, or be good or be bad. We'll see that all in the same mutation here. So this is on sickle cell anemia, and I'm going to play a short video first.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
What we got here, guys, is here's normal, right? Just a single letter change from a T to an A here changes just a single amino acid in that hemoglobin protein, but that changes the way it behaves. Here, you got normal hemoglobin, blood flows just fine, no problems. Over here, you have sickle cell anemia. Some of the blood cells will sickle, get stuck, cause pain, and can even cause earlier death, usually not until later in life. So it seems like it's a bad mutation, but there are some cases that we'll talk about when we get to genetics that is actually a beneficial mutation in certain environments, but I'm not going to get into that right now. We're going to foreshadow it so you have something to look forward to in the future. Okay, guys, so that kind of covers mutations. Very simply, a change in the letter order in the nucleic acid order in DNA gets transcribed into mRNA and then translated into a slightly different protein, which could have no effect, some small effect, either positive or negative, or some major effect, either positive or negative for you. They don't happen often, but when they do, they might have all those different effects. And that will cut it for us on notes for our DNA and protein synthesis unit. Do you guys also want to add?
Yeah, I like to leave the students with actionable advice. You know, this is all related to life, and so how can we apply this to everyday life? And I think most students have probably picked up on this already, but it isn't wise to expose yourself to gamma rays or radioactive spiders in order to become Hulk-like or Spider-Man-like, because mutations are random. Most of the time, they're not beneficial. It's easier to screw something up than it is to make it work better. So hold off on exposing yourself to things that cause mutations in the hopes of gaining some sort of superpower. More often than not, it does not work. Let me tell you from experience.
Yeah. And that goes for things that don't give you superpowers too, like increased sun exposure, tanning, smoking. Those are all things that can cause mutations or carcinogens.
Mm-hmm.
You know, I haven't noticed it as much recently, but tanning used to be such a trend, and that was something we used to talk a lot about when I was student teaching with you too. We did the integumentary system. So,
Mhm.
yeah, that was a real. I don't know if it is still a thing. Do people still like to do that a lot? Don't do it intentionally. It's
I don't.
Not good for you. The more you know.
all right. You need that. I hope you find that sound to add in and editing. Make your work longer. All right. Should I hit stop now?
Bye,
Please.
guys.
Probably.